Hello everyone. One of the most common questions that I get when asked if we could do practical in a lesson is the extraction of DNA. It's one of the most fascinating things to do and to visualize. And using a single strawberry, you can actually extract a sample of DNA and have a look at it without even using a microscope. So this video is just a bit of a tutorial just to talk through how you can actually use certain equipment to get this DNA, this deoxyribonucleic acid, how we can actually visualize this. So I'm just going to talk you through some steps, and I've got at the top here, just in a sort of bullet point form, the kind of ingredients or the equipment that you're going to need to do this. So if you get these particular pieces of equipment, you'll be able to follow this method. So the first thing we want to do, I'm just going to put step by step what we're going to have here, is we're going to want to measure out about 90 milliliters of water into a beaker. So we need to get a beaker and put into that about 90 milliliters of water. So we've got 90 milliliters of water. Now into that beaker we're going to add 10 milliliters of dish soap or washing up liquid. So we need to take a bottle of washing up liquid. I'm just going to keep this fairly visual just by doing a few little pictures. So we've got some washing up liquid or dish soap, but we need to add that into the water. So that's about 10 milliliters of the dish soap. And I've got the values at the top there too. Now also to that we need to add 1/4 teaspoon of salt. So we're going to have a little container of salt, and we want to add that to our water also. And I've got at the top here just the value, so we want 1/4 teaspoon of that. Now what we need to really do is mix it all up. So essentially what we're forming is a homemade extraction solution. So we're going to mix all that up. So I'll just put here number 2. Very simply mix it all up, and that's going to produce a homemade extraction solution. Now then what we need to do is take our strawberry and put that into one Ziploc bag. You know, the kind of small freezer bags that you can get, where at the top there's a sort of little dashed bit, a bit like this, that you can seal it up. So we're going to get a Ziploc bag, and we're going to place into that Ziploc bag one of our strawberries. Not the best picture, but there we go. Add a few of the pits on. There's our strawberry. And this here is our zipper lock bag. Now if you can't find one of those, any kind of bag with a seal at the top would work for this. So we put the strawberry into that Ziploc bag, and then what we need to do is pour the extraction solution that we've made into that bag. So I don't know why I put it in purple there, but we're going to pour our extraction mixture into, and I'll just put into the zipper bag. There. Now when we do that, what we want to do is remove as much air as possible, and then we want to seal it. Once we've sealed it, so once we've poured our extraction mixture into that Ziploc bag and we've sealed it tight, you want to use your hands to really mash, smash and mush essentially the strawberry inside of the bag until there are no large pieces remaining. So number 2 we had mixed it up. For number 5 this time we're going to have mash it all up. So we don't want any large pieces remaining. Now once we've done that, we're going to pour the resulting strawberry and extraction solution mixture through a sieve into a beaker or a similar container. So what we're going to do for step 6 is we're going to take a sieve here. I'll just draw a bit of a mesh there just to remind you. So we've got a sieve there. Here's our beaker, and we're going to essentially pour. So if I draw a bit of an arrow from this top picture of the zipper bag, So we're going to essentially pour in our strawberry extraction mixture through the sieve into a beaker. And we're going to start to collect fluid in the bottom of this beaker. Now what we can do is use a spoon, and I think we've got a spoon at the top here, so we've got a spoon in the apparatus list. I'm going to use a spoon to press the strained bits of strawberry against the sieve, just to force even more of the solution into the beaker. Now I've put here that from the container it's currently in, from this beaker, we could transfer the solution into an even smaller beaker or a similar container that holds around 50 to 100 milliliters of fluid. We don't essentially have to, but what we're saying is we only really need about 50 to 100 milliliters of fluid. So for the next bit, that's all that we're requiring to essentially put through the sieve. So this will work with just 50 to 100 milliliters of fluid. Now what we want to do once we've got that fluid is add 5 milliliters of our chilled isopropyl alcohol or ethanol to the solution. We want to hold that mixture at eye level. So at the top here, I've got that we're needing 5 milliliters of isopropyl alcohol. And what we're going to do is, if I just put that with an I to represent that isopropyl alcohol, what we want to do is just pour that in to this beaker there. Now we're only wanting, again, 5 milliliters of that. 5 milliliters. And usually it's good to drop it down the side. So it's nice to drip that isopropyl alcohol down the side of one beaker rather than just pouring it straight down into the middle. Now if we hold that at eye level, what you'll notice is that there's this separation of what appears, usually the response I get from my students is this white stuff on top of the rest of the solution. Now if you wait about 30 to 40 seconds, you see this white kind of layer forming, looks a bit fuzzy. That white layer is the DNA. That is the DNA of the strawberry. Or we can use the tweezers that we've got in our apparatus list. So we can use these tweezers to actually pull that DNA out. Now clearly, just because we're looking with the naked eye, we're not going to be able to see the double helix structure that you classically think of when we think of the DNA. So we're not going to be able to see that, but you can physically have within these tweezers the actual DNA from the strawberry. And there you have it. It's a very simple, very quick method of extracting DNA that you can do in a lab. The apparatus to get hold of is very quick, very straightforward. Very similar as well. You can use, this works with a Kiwi. You get equally nice results. I know certain methods that you can research on the internet use a blender to mix things up. Personally, I'm not a fan of those. It's a bit more involved, takes a bit longer. This you can really pull off in about 10 minutes, 10 minutes max. So all we do is we take a beaker, put in the water, salt, dish soap, we mix it all up to get an extraction solution. We add it to a strawberry in a Ziploc bag and really mash that up. And what we're essentially doing is breaking down the cellular components to expose all of that DNA. We're going to sieve that mixture through to just get a clear liquid into another container. We're going to use a sieve for that. And to 100 milliliters of that extraction fluid, we're going to add 5 milliliters of isopropyl alcohol, and we should be able to get a nice bit of DNA forming. Okay, hope all that helps.